Welcome all of you. This is Professor Suraj Adole. We are going to start with new topic and enzyme. Enzymes. So, enzymes study is called as enzymology, a substance produced by a living organism which act as a catalyst to bring about a specific biochemical reaction is called as enzyme. So, we have discussed some content in this topic. First is chemistry of enzyme, then classification of enzyme, then mechanism of enzymes action, then enzyme kinetics, then some inhibitory factors of enzymes, means some inhibitions of the enzymes we are going to study here, some activator factors of the enzyme we are study here, and the specificity of the enzyme. So now start with chemistry of enzyme. Here introduction. An enzyme are the biological catalysts that speed up the rate of biochemical reaction. Here listen carefully. It is a biological catalyst. Means enzyme also called as catalyst. So enzyme act as a catalyst for specific chemical reaction, converting a specific set of reactant which we called as substrate into specific product. Without enzyme, life as we know it will not exist. So enzyme are the protein molecule in the cell which work as a biological catalyst. So enzymes are protein, but all proteins are not enzyme. We are already know about that. It was study in 12th standard board syllabus. So most enzyme are the three-dimensional globular protein. So here most of the enzyme are the three-dimensional globular proteins. So protein have a nature which are tertiary and quaternary structure. Means enzymes are always present in tertiary and quaternary structure. Here see there are four structure of protein. First is primary structure of protein, then secondary structure, then tertiary structure, and last one is quaternary structure. Then we have studied some special RNA species also act as an enzyme called as ribozyme. Here we discuss the diagram of hammerhead enzyme. See, it is three-dimensional structure of hammerhead enzyme, which is tertiary or quaternary structure of this enzyme, which we called as ribozyme. So here, characteristics of enzyme. Enzyme, first characteristic of enzyme is enzyme speed up the rate of the reaction. Means it speed up the rate of the reaction by lowering the activation energy. Means here activation energy is lower, or which we call as transition state is also lower. So their presence does not affect the nature and properties of enzyme. Means enzyme presence does not affect any product. Means in the presence of enzyme some different product are there, and in the absence of the enzyme some different product. This was not happen. They are highly specific in their action. That is, in each enzyme can catalyze one kind of substrate. Means enzyme are highly specific. Means one enzyme recognize one substrate only. The small amount of enzyme can accelerate a chemical reaction. Enzymes are sensitive to pH and temperature. Means enzyme are work in optimum pH and optimum temperature. If you increase some temperature, then enzyme will not work properly. Here turnover number is defined as the number of the substrate molecule transformed per unit by one enzyme molecule is called as turnover number. Means here catalyst turnover number equals to 6 into 10 raised to power 6 per minute. Means 6 into 10 raised to power 6 substrate are converted into 6 into 10 raised to power 6 product per minute in the presence of one enzyme only. So structure of enzyme. The enzyme are also called as hollow enzyme, which cover enzyme part plus cofactor part. Here, in the absence of cofactor, enzyme is also called as apoenzyme. Means without cofactor, enzyme has no role. Means enzyme can combine with cofactor, then it will be active form of the enzyme. So in that enzyme, there are some active site of the enzyme in the region where substrate can bind, plus cofactor also bind, and prosthetic group is also bind, and contain reduce that help to hold the substrate. The active site of generally occupy less than 5% of total area of the enzyme. Means out of 100% enzyme part, only 5% are covered by active site. So active site has a specific shape due to tertiary structure of the protein. Change in the shape of the protein affect the shape of the active site and function of the enzyme. Next slide. Active site has a two part. One is binding site and another is catalytic site. Means binding site will bind to the substrate and catalytic site will catalyze further reaction. The active site has also bind to allow cofactor. Means without cofactor, enzyme is non-active. We are already studied that. So how many type of cofactor are there? There are some cofactor which has non-proteinous molecule which carried out chemical reaction that cannot be performed by standard 20 amino acid. Means always listen that ki cofactor are has non-proteinous molecule. So there are two type of cofactor here: organic cofactor, inorganic cofactor. So organic cofactor and inorganic factor. First we study this image. Here cofactor will bind to the protein, means enzyme. Here see that cofactor is bind to the enzyme, then it will be the active active protein. Here active protein. If cofactor is not bind to the protein, then this protein will be active. Here protein is nothing but enzyme. So inorganic cofactor, there are inorganic molecule required for the proper activity of the enzyme. Example here inorganic cofactor are carbonic anhydrase. Carbonic anhydrase is an enzyme where Zn dipositive is an inorganic cofactor. Zn dipositive is inorganic cofactor. So organic cofactor, there is another example of inorganic cofactor, hexokinase. We already know about that. Hexokinase is an enzyme in glycolysis pathway. Hexokinase is the first enzyme on that pathway. This pathway is 10 step. Hexokinase has a cofactor of Mg positive. Mg positive also a cofactor of DNA polymerase. So organic cofactor are here. There are the organic molecule required for the proper activity of the enzyme. Example, glycogen phosphorus required a small organic molecule, that is pyridoxal phosphate. So here glycogen phosphorylase is an enzyme which involve in gly glycogenolysis, which are involved in glycogenolysis means glycogen breakdown. So it is rate limiting step of glycogen breakdown or glycogenolysis cycle. So it required a cofactor which is called as pyridoxal phosphate. This glycogenolysis are break down the glycogen and convert this glycogen into glucose when the blood glucose level is low. So another slide here. Type of cofactor. Type of organic cofactor. Here are two type of organic cofactor. One is prosthetic group, another is coenzyme group. A prosthetic group is a tightly bound organic cofactor Example, flavin, heme group, and biotin. Here is hemoglobin chain, where heme group is bind. There is two chain of beta and two chain of alpha, because hemoglobin is a tetramer in structure. Okay. Here iron part means is here heme group part, which we see that it is a prostatic group of organic cofactor. Then coenzyme group of organic cofactor. Coenzyme is loosely bound to the enzyme. Here listen carefully. One prostatic group is tightly bound to the enzyme, and another coenzyme is loosely bound to the enzyme. And this is difference of prostatic group and coenzyme group. Coenzyme group is always loosely bound to the enzyme, and prostatic group is tightly bound to the enzyme. So the enzyme with cofactor remove is digested designated as apoenzyme. Sorry, the enzyme with cofactor removal, we already discussed that without cofactor holoenzyme is called as apoenzyme. Without cofactor, the complete complex of the protein with all necessary small organic molecule, that is metal ion and other component, term as holoenzyme or holoprotein. We have already discussed that holoenzyme is made up by enzyme plus cofactor. This is a binding of substrate to the enzyme, and the reactant in biochemical reactions term as substrate. When the substrate bind to the enzyme, it form an enzyme substrate complex, which was later on converted into product. The site of the enzyme synthesis. The enzyme are synthesized by ribosome, which are attached to the rough endoplasmic reticulum. As earlier we studied about the molecular biology, in that we study the protein synthesis is a site of endoplasmic reticulum on which ribosome unbind, and we already discussed about that all enzymes are protein but all proteins are not enzyme. Then through this statement we referred as enzyme is nothing but an protein. So protein biosynthesis occur in endoplasmic reticulum. That's why enzyme biosynthesis also occur in endoplasmic reticulum due to the help of protein synthesis factory called as ribosome. So here information of the synthesis of enzyme is carried by the DNA. Here is the first step where the enzyme information is on the DNA, which was copied on the mRNA, which we call as transcription, and this copied mRNA will convert it into protein, which we call as translation. And this protein synthesis are done on endoplasmic reticulum with the help of the ribosome. So amino acids are bonded together to form a specific enzyme according to DNA code. So there are two type of enzyme, broad classification here, intracellular and extracellular enzyme. So intracellular, this is enzyme are work inside the cell and synthesis also inside the cell. The enzyme are synthesized and retained in the cell of the uses of the cell itself. They are found in the cytoplasm, nucleus, mitochondria and chloroplast. The example of intracellular enzyme are so much. One of the example is enzyme involved in reduction in the mitochondria. All enzyme which was used in reduction in the mitochondria are intracellular enzyme. There are also some enzyme which was present in the nucleus, which is called as DNA polymerase, RNA polymerase, ligase, topoisomerase. Lot of enzyme of intracellular enzyme. So there are some extracellular enzyme. The enzyme which are synthesized inside the cell but perform work out of the cell, means their functional activity is beyond the cell or outside the cell, is called as extracellular enzyme. So digestive enzyme mostly produced by the pancreas are the extracellular enzyme. There are also a lot of, and their work to transport the duodenum. So another side, nomenclature of enzyme. The enzyme are named according to the name of substrate, its catalase. Some enzyme were named before the systematic way of naming the enzyme was formed. Example pepsin, trypsin, name. The, by adding the suffix ASE at the end of the name of the substrate and the enzyme are named. There are a lot of example which has there are a lot of example of the enzyme which has suffix ASE. Here you see maltase, maltose and water. In the presence of water, with the help of the enzyme maltase converted into two molecule of glucose. Here see two molecule of glucose. Means maltose is a disaccharide which are break by the enzyme maltase. There are also some enzyme which does not has suffix ASE but they are act as enzyme like pepsin, trypsin, renin, which we already studied. So there are some example of the enzyme which have suffix ASE. So here this example lactase, sorry, lactase, maltase, cellulase, lipase, amylase, protease. So lactase is an enzyme which break the lactose. Lactose is also disaccharide sugar like maltose which convert glucose plus galactose, but maltose convert two molecule of glucose. Cellulose is a homopolysaccharide present inside the cell wall. So means cellulose is made cell wall, but it will break by an enzyme called as cellulase. This cellulose has has a bond in between two glucose molecules. This bond is beta 1,4 glycosidic linkage, and it was very tough to break. But only cellulose is an enzyme which break this cellulose. There are also lipid molecule break by an enzyme called as lipase. So lipase converted into lipid into glycerol plus fatty acid. There are also amylase which break down the starch into maltose. There are also protease which break down the protein break down into peptides and polypeptides. So classification of enzyme. The enzyme are classified by a International Enzyme Commission. This classification on the type of the reaction catalyzed by the enzyme. There are six major classes which we shortly recognize as an othal. The enzyme class is further divided into subclass, and this class is further divided into subclass which is called as subclass, and soon to describe the huge number of different enzyme catalyzed reaction. So first enzyme here oxidoreductase. So reaction type is reduction and oxidation carried out. So example here is lactase dehydrogenase. So lactase dehydrogenase is an enzyme which carried both reduction and oxidation. Due to this we call as oxidoreductase. Here see first, this is an enzyme classification. There are six type of enzyme which we recognize as othal, O T H L I L, othal. Okay, so shortly you can recognize from this name whole classification of enzyme. So here we first study oxidoreductase. Second one is transferase. Transferase is a move chemical reaction. Example hexokinase. So sorry, chemical reaction, not chemical group. It's move the chemical group. Here hexokinase is help to move the chemical group. Means here ATP has a phosphate group which was moved from ATP toward the glucose, and glucose is converted into glucose 6 phosphate with the help of this enzyme called as hexokinase. Then second one is hydrolase. Hydrolase is an enzyme which was work in the presence of water and which was helped to break down some chemical bond, and it also help to transfer some group also. So here is an example of this hydrolysis, lysozyme. So another class is lysis. It is a non-hydrolytic bond cleavage, means without the help of water the cleavage is there, is called as lysis. It is a good example of fumarase. Here fumarase is present inside the Krebs cycle, this enzyme, and it help to cleave some bond. So another class is isomerase. Now there are also one example of lysis, the fructose base phosphate aldolase. Means here aldolase is converted fructose 1,6 bis phosphate into two molecule, that is 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. So another class is isomerase. So this class are intramolecular group transfer, that is isomerization, that is triose phosphate isomerase. This enzyme are work in glycolysis cycle where glucose 6 phosphate is converted into fructose 6 phosphate. So last class is ligases. So here ligases are work in the presence of ATP. They synthesize the new covalent bond. So here example of RNA polymerase. There are also one example of